Hi, my name is Evgeniya, and I am doing my PhD studies at the Life Sciences Department of Chalmers. I study the biophysical characteristics of DNA-protein interactions from a single-molecule perspective and work on developing and improving single-molecule techniques. In this talk, I will introduce some of them and explain why we want to use them. Let's start with a little bit of biology. All biological organisms are made of one or more cells, making cells the building blocks of life. Cells, in their turn, are comprised of biomolecules, such as DNA and proteins, that participate in many biological functions and are heavily associated with each other. DNA stores genetic information, and proteins maintain DNA and basically do everything surrounding the functioning of the DNA. For example, proteins play a role in genome maintenance, where they help store DNA and protect it and make it available or unavailable for other processes. Proteins also play a role in transcription. When DNA is used as a template to produce RNA, proteins are the machinery of that production. Same with the DNA replication. Proteins participate in duplicating DNA copies and protecting DNA during that process. Proteins also drive the DNA repair. DNA can get damaged, and that's very detrimental for the organism's survival because, as I said earlier, genetic information is written on the DNA and the organism wants to keep it intact. So, DNA breaks must be repaired, and proteins are the ones that do it. These processes that we've now discussed are just examples. There are so much more things happening in the cell. These examples are fairly well studied, but there is still so much more to learn on a deeper, more detailed level. That's what we're trying to do with single-molecule methods. But what exactly do we want to learn? What characteristics do we study when we look at DNA-protein interactions? For example, we can try to see whether the protein and DNA bind strongly between each other, like Velcro steps on your sneakers, or if the binding is weak, like a post-it note sticking to your notebook. That describes the binding strength or binding constant. We can also investigate whether there is a specific part of DNA that the protein wants to bind to. Let's imagine our protein as a magnet of a certain polarity, north, for example, and a part of DNA being of the opposite polarity, south, while the rest of DNA is not magnetic. North magnet can only attach to the south one, so the protein will look for that particular part of DNA, and when it finds it, it will bind. Alternatively, the whole DNA can be available for the protein to bind, like a fridge to the magnets from your trip. This is what we would describe as a specificity of the binding. Another thing we can look into is the behavior of the protein when already bound to DNA. For example, proteins bound to DNA can behave like beads on a thread, rolling along the DNA. Alternatively, some protein-DNA complexes can act like a spool of thread. It means that DNA thread can wind onto that spool of the protein. These are the examples of the dynamics of the DNA-protein interactions. All the processes that we discussed before, such as transcription, DNA repair, and others, can be characterized by these categories: the strengths of interaction, the specificity, the dynamics, and so on. And this is what we want to learn about these processes. Why do we want to learn it? Well, as I also mentioned earlier, DNA-protein interaction is fundamental for most biological processes. For example, let's say some process in a human organism isn't working exactly how it should. If we know how it should work, we can find drugs that will help fixing the process. Alternatively, if we know exactly how some process works in a pathogenic organism, we can develop a drug which will disrupt that process, that way killing a pathogen. Now we know what we want to study. We know why we want to study it. How do we study it? There are many biophysical and biochemical methods available for DNA-protein interaction studies. They can be divided into two groups: ensemble or bulk methods and single molecule methods. To visualize it, imagine that we have a flask full of sheep with black and with white wool. If we want to analyze what kind of sheep are in the flask, we can use a bulk method. From it, we will get some information, maybe about the size of the sheep, the fluffiness of their wool, but some information can get lost in the averaging process of the bulk assay. For example, in this case, we would see that the sheep in the flask are gray, which is not entirely true. And this is where single molecule methods come in. With the single molecule techniques, we can take each and every individual sheep, look at it closely, and analyze it. This can help us figure out that we have both white and black sheep in the flask. We can also do some statistics to find out how much of each color is there, cross-correlation analysis, and so on. Obviously, this example I'm giving you is very, very simplified, and there are much more complications to the process. And there are pros and cons to both bulk methods and single molecule methods. But the main pro of single molecule measurements is that you can view individual molecules, and that way you can enhance the knowledge from the ensemble methods. There are many different single molecule techniques which differ in the readout, in conditions of the experiment, and in the methods used to stretch the DNA sample. You can check out the video description if you want to read about some of the methods. For example, there is a method called optical tweezers. The DNA is bound by its ends to the beads, and these beads can be moved so that DNA will be stretched. The resulting readout is a graph that will look different depending on the structure of DNA, which in turn can be affected by the bound proteins. The downside of this method is that we cannot visualize the molecules and that the ends of the molecules must be occupied by the beads. Another example is a group of microscopic methods such as atomic force microscopy or scanning electron microscopy, where the sample is deposited on the surface and scanned on top. This produces very nice, high-resolution images of the DNA molecules. The downside is that the sample preparation is usually rather complicated and the conditions are not physiological. But what if we want to have a DNA sample where the ends are free, nothing is attached to them, DNA is in solution in more physiological conditions, and we can still see the individual molecules? Well, we can use fluorescent microscopy for that, but we would need to combine it with some sort of DNA stretching to be able to see individual DNA molecules better. And to do so, we can use single molecule nanofluidics. Single molecule nanofluidics is the main method we use in our group. It is based on stretching the DNA in nanochannels due to confinement. To stretch DNA, we utilize a custom-made nanofluidic device that you can see in the video. Two of the devices are set on the same chip, and if we look closer at one of them, we can see that it consists of 4 wells. Each pair of wells is connected by the microchannels, and the microchannels are connected by nanochannels in between. The sample we usually study consists of DNA, which can be mixed with a protein in solution. The DNA is usually fluorescently labeled, which will make it visible under a microscope. For example, one of the most common dyes we use is called Yo-Yo-1. Let's look a bit closer at the nanochannel entrance. When we load the sample in solution in the top left well, and then apply the nitrogen gas pressure, the liquid is pushed into the microchannel. That makes the DNA move through the microchannel. When we then apply the pressure to both of the top wells, the sample is pushed down and the DNA goes into the nanochannels. Combining the stretching with fluorescent microscopy, we image the DNA molecules. You can see real DNA molecules that are fluorescently labeled and stretched in the channels, wiggling in the video. To explain why we need stretching and make the process a little bit more visual, imagine the sample I was talking about, the DNA in solution, as a bowl of noodles. And of course, when you have a bowl of noodles, you cannot see what's happening to an individual noodle. All the strings are chaotically mixed, tied up, and overlapping. But if you take the chopsticks and lift and stretch one of the noodles, then maybe you can see that it, for example, has a knot on it. These knots, or other unusual structures on otherwise straight and even DNA noodles, is what I am looking for in my experiments. These structures are usually associated with the effects of the proteins. Additionally, I would like to mention that if we use the same types of channels and the same conditions, we get a very uniform stretching. It means that the extensions of the DNA molecules on our images represent the real lengths of DNA molecules. And thus, we can compare different samples with or without protein, or with different concentrations of the protein between each other. This method allows us to visualize DNA-protein interactions in solution, close to physiological conditions, to investigate individual acts of protein-induced changes in DNA conformation, as well as gather significant statistics on various protein-associated processes on DNA. Now you know a little bit more about the DNA-protein interactions and ways to study them at a single molecule level. Hope it was interesting. Thank you very much for listening to this talk.